Again, thank you so much, Martha, and to all the organizers for allowing me to uh, present today. Hello, everyone. So I will be presenting a pre-recorded lecture because of the weather here now. I am not sure if I will be. I might be having some technical problems. So let me just. Can you see my screen already? So let me start with the presentation.
Yes, we can.
Okay, thank you. So good day, everyone. I'd like to thank the organizers of the conference for allowing us to present part of our work in our laboratory. So today I'll be showing to you part of our results. So we did our study, the polyphasic survey of Aeromonas hydrophila phages and genomic characterization of novel lytic phage, Aeromonid phage B614. This is one of our initial efforts in our laboratory here in Metro Manila in the Philippines to describe the diversity of phages in our collection. This also employed basic physiological and genomic characterization of the phages in the country, obtained from different samples. And this paper is also being presented as a poster paper in this conference with my co-authors, Tracy Gutierrez and Ruel Bennett. Most of the studies done here in our country has focused on the isolation and some application of phages, ranging from aquaculture and even to the food industry. Characterization commonly performed is through morphology, which only, of course, allows morphological classification of the isolates. However, there's a need to further the characterization of these phages so that we can, of course, fully understand their nature, diversity, and their ecology. Here are some of our papers that we have done here in the lab, looking at their application on Oreochromis niloticus infection, and we also looked at some of their comparison of their delivery systems also in Oreochromis niloticus. Okay, so this is a map of the Philippines, and we isolated the 27 phages that were used in the study in the largest island in the Philippines, which is Luzon. So this is now the map of Luzon, and we were able to isolate 9 phages from sewage samples in Manila, and 9 phages also coming from different lakes. Lake Taal, which is found at the middle part of Luzon, and Lake Taal actually houses a volcano, which is a very interesting environment for phages. And then we also isolated phages from Lake Paway in the northern part of the Philippines and Laguna de Bai Lake, which houses a lot of fish cages and a main source of income in the region. And then we were also able to isolate 7 phages from a river system in Mikawayan, Marilao, and Obando in the province of Bulacan, and also 2 phages coming from an aquaculture pond in another province known as Pangasinan. So some of the phages were actually already initially used for therapy, as you can see here, and what I have mentioned a while ago in some of our publications. But we also aim to classify the phages first using gene 23. So we wanted to ultimately determine if there is a congruency between the physiological and genomic data of the phages. The relation between the phages characteristics and their isolation source were also considered in the study, together with the marker gene analyses and to gauge the diversity of the phages in the collection. So showing here to you now are the different phages that we used in the study. So these are now a total of the total or the morphology of the different phages that we used in the study. Okay, so with the main objective in mind, we have designed the study in order to obtain polyphasic characterization of the phages. Phages were acquired from our culture collection in the beets collection from our research group, and the phages were propagated using Aeromonas hydrophila PNCM 10089 strain. And after propagation, eventually the phages they underwent stability testing. Classification and phylogenetic analysis of the phages were done by amplifying and sequencing a signature gene from the extracted phage DNA. Additionally, previous data on the whole genome analysis of phage B614 was used and also analyzed in this study. So a collection of parameters were used to check the physiological and lytic characteristics of the phages and were summarized based on their original isolation source. This includes host range, pH and temperature stability, as well as stability in various saline concentrations. So from the results, it was observed that the phages isolated from the sewage had the most diversity in their lytic abilities, which can be explained also based on the nature of the habitat, which provided a conducive environment also for the host bacterium, thus enabling frequency and the exposure of the phages to the host. So you can see here with the different parameters that were used, also in the host range, they were still able to infect some of the hosts that were tested against them. And for the pH, all of them were stable at pH 7, but some were tolerable from pH 5 and also up to pH 9. And then for the temperature, most of them were stable at 40 degrees and 25 to 30 degrees. Depends on their environment, and also their sensitivity to salinity. They can still infect their host even after their exposure to high saline environment, but already showed substantial decrease in the capability of infection by the phages. So this is now the list of the different phages that were tested and just some of the results that we obtained from this characterization tests. Hierarchical clustering of phages using physiological data sets were performed. So here, phages in the same branching are treated as same species, equating to one viable phage isolate. So in our study, the method showed that although a small sample size was used, a high diversity of the phages were still observed in the collection. So the number of viable bacteriophages, or in this case, phages with varying sets of characteristics, was reduced to 60%. So this, as well as the varying abilities of bacteriophages, actually present important information, especially if phages were to be employed in various applications. In vitro testing of phages can be done in optimal conditions. However, in vivo experiments can be prone to suboptimal conditions, which may sometimes lead to unsuccessful applications of these viruses, of these phages. So knowing the degree of the diversity and the abilities of the bacteriophages can be used and can help researchers evaluate which phages can be used in different conditions. Moreover, it should be noted that these phages were isolated from different environments. So now for this study, our results show the hierarchical clustering presented. It presented actually no grouping according to the sample area. So we have the sewage, the lake, the river, and even the aquaculture pond. So this may present actually or we may conclude that this presents a local microdiversity of these viruses from the four sample types that were mentioned. Though there were more than 20 phages that were used in the study, we first looked at phage B614 isolated from a sewage sample as the first phage that with its whole genome being sequenced. We will of course work with the other phages too, but we initially look at B614 first. So through proteomic analysis and determination of the average nucleotide identity between reference phage ASF-D1 and phage B614, the novelty of the isolated phage was actually observed. So based on published species demarcation of 95% for bacterial viruses, it can be suggested that phage B614 is a novel phage species belonging to the Bicortoviruses due to its 84.2% an average nucleotide identity with the known reference phage. So we know that, of course, Bicortovirus is a genus under the tail bacteria Phages represented by 44RR 2.8 T phage. But ASFD1 phage was used as reference in the study instead of the type species because it showed the highest similarity based on GenBank analysis. So this is now the phage B614 showing you now the micrograph of the phage B614 and its genome. So B614 was used as it is part of the collection here in our laboratory in Manila with the whole genome sequence, as the present study is limited only to the analysis of Philippine phages. B614 was used as a representative of all the Philippine phages for now. From the results of B614, probable taxonomic classification of the other isolated phages were determined. So the phages showed less than 85% average nucleotide identity to reference phage ASFD1 and less than 95% to phage B614 based on major capsid gene sequences. So showing that the other Philippine phages are more closely related to the probable new species B614 than to other reference phages in the GenBank when the major capsid gene is analyzed. So this clustering may suggest a novel species of phages under the genus Bicortoviruses based on gene 23. In this case, conspecific relation of the said phages may be suggested. So this grouping can be attributed to dispersal limitation as well as environmental conditions, which can be actually related to the environmental conditions and geographical isolation of the area. So through analysis, with the use of the marker gene, it may be suggested that the group of viruses present a group of novel species of Bicarta viruses. But, however, further analysis of the phage genome, the whole genome, similar to what was performed to B614, should be done to establish the novelty of this claim of the other phages in this study. So it is vital in order to provide an accurate taxonomic classification and identity of the other 20 phages, Aeromonas phages, that were used in the study. Just to summarize, so microdiversity of phages is evident in the results of this study, indicating isolation of phages from same environment does not necessarily imply their evolutionary relationship based on our results. Another factor to be considered is the geographic diversity of the phages. This may be influenced by the archipelagic isolation of the country, the Philippines, which contributes to dispersal limitation and the unique climate that dictates environmental conditions. So eventually resulting in the unique grouping observed in all the clustering methods performed. Although to further study on aeromonad phages in the country is recommended to establish this novelty. This study provides a first survey actually of the diversity and identity of phages in the collection here in Manila. So along with this, ahydrophilophages isolated from various sources in the country or environmental samples in the country also clustered separately from the reference phages that were used. With the initial characterization of B614 phage and the cluster shown in the phylogenetic tree, it may be deduced that phages belonging to an unclassified group of Picorta viruses. So with that, I'd like to thank everyone for listening. Again, thank you to the organizers, and I'd also like to thank my students from our laboratory and the different institutions who made this study possible.
Thank you very much for that very interesting presentation on those pages. Are there some questions from the audience? I can perhaps start off the questioning. I wanted to ask you about the, because you had all the clustering based on phenotypic observations. So how different are the phages in terms of their host range and their other properties that you looked at? Do they seem quite variable?
Yes, for most especially for the phages from the sewage samples were actually um, they were their activity and their infectivity to their host were actually let's say higher, or they're more active. And then the other phages that were isolated, like example from the river systems, were those that would just be stable at the typical let's say temperature. And if you expose them to let's say more acidic or more basic or higher temperatures or more harsh environments, then they already decrease the infectivity. So yes, for I think that yeah, that's why also we chose B614, because from the different results that we had, and even the host range, it was really able to infect a lot of not just the mutants but also other Eremonas that we had in the lab.
Interesting. And then also I was wondering, because you used the GP23 as a marker, so did you see other phages that didn't amplify with that gene? So have you got other sort of families of phages in there as well?
Yes, I think there were a few of the phages. Yeah, there are a few phages. I think uh, there were at least 7 phages. I think yeah.
It's interesting that so many of them were from that related complex. Okay, well, thank you again for sharing the data with us. That was a really excellent presentation.
Thank you.